If we talk about the details of the DNA replication process, we are going to be starting out with a double-stranded DNA molecule. Now, within this double-stranded DNA molecule, there will be certain areas where this whole process starts, and these are going to be known as origins of replication. And so those origins of replication, there will be many of them, and this would be per chromosome. And when we say many, we're really talking about hundreds of these. And so having a lot of them is going to help us just to accomplish this whole process a lot faster, because we will have a lot of DNA that has to be copied. And at these origins of replication, specifically what is going to happen is we call it melting the DNA. But what that means, melting the DNA, we are separating the two strands. Okay, so we're going to have localized areas where the two strands are going to actually separate from each other. And to get even more specific about that, really the hydrogen bonds that are holding the strands together will be broken. Okay, so as soon as those are broken, the two strands will separate from each other. Again, it's just going to be a localized area. So if we're just looking right here, maybe it's just this little area right here that's going to actually open up from each other, because all of those hydrogen bonds that will be on the inside here, we're just going to have a specific enzyme really that's going to go through and actually break those so that we can separate the two strands from each other. And when that happens, we're going to see that we get this little bulge in the DNA where those two strands separate. And we're going to call this separation, this little area, we're going to call this a replication bubble. Okay, so let's take a look at that once we have this bubble actually formed. So as we look at it now, we can see that our two strands have separated from each other in this localized area, which again is what we're going to call the origin of replication. This would be occurring all over the DNA strands. Now, the enzyme that's responsible for creating this bubble is going to be called helicase. And so all of our enzyme names, I will point out that they end in ase there at the end. So this is really just the first one for us. So what helicase is going to be doing is it's actually kind of bulldozing, if you will, its way through here. And so we will have helicase operating on both sides of this. So we'll have one on each end, and helicase is responsible for breaking the hydrogen bonds that were holding the two strands together. Now, where we see it actually drawn on here, just to give you an idea of some terms, this area where it's drawn on, this would be called a replication fork. It's like a fork in the road, and then we have another one over here. So within one replication bubble, we're going to have two replication forks. And we will really have a whole set of enzymes working at each one of those replication forks. Okay, so helicase is actually separating the strands for us. Once these strands are separated, they do want to reconnect with each other. So we would call that process reanneal. So that means they want to stick together because, again, the G's and the C's, for example, they could form 3 hydrogen bonds and they're still present there, and the T's and the A's, they still want to form the 2. So we have to have a way of preventing those from actually reannealing with each other and reforming that double-stranded molecule. So we will have other proteins that are going to be helping to keep the two strands really propped open from each other. So the proteins that are responsible for doing this, they are going to be located right here. And so we will see them really on both sides of this. And they are coating those bases that are now exposed. And so these proteins, as I've just drawn them, these are called single-stranded binding proteins. And specifically, these are going to prevent the strands from reconnecting. So they're not going to allow those hydrogen bonds to reform, because at the moment we don't want them to reform, because we're going to be building the new DNA directly on top of this old template DNA strand. So the black molecule, as we go through this and we use the word template, template is the old DNA. Okay, so shown in black in this picture. So once we've got the two strands separated from each other, we're really ready to begin this whole process, and we can see the bases. We have enzymes that will be able to read those and interpret, and they know the base pairing rules. So if they see a T, they'll know to put an A across from that, and so on. First thing that has to happen though is we have to have a molecule to really help get us started in this. So if we talk a little bit about the enzyme that's going to be responsible for actually making the DNA, that enzyme's name is DNA polymerase. DNA polymerase is oftentimes just called DNA pol for short, and DNA polymerase, it will be responsible for reading the template DNA, and it's going to actually be making the new DNA strand. And so as it does this, it really is connecting nucleotides together. And so when it connects those nucleotides together, it would be actually making phosphodiester bonds. So for example, if we had a DNA strand here and this was the sequence, then DNA polymerase would be able to look at the A and know that across from that goes a T. And then it could look at the next one and it could know that across from a T goes an A, and so on. So it will be able to interpret that and then incorporate the correct bases based on the base pairing rules. However, there's a few specific things about DNA polymerase, or in other words, things that it can't do, which does affect the way that we have to replicate the DNA sequences. So first off, DNA polymerase cannot start the synthesis, or we could say initiate DNA synthesis on its own. So what that means overall for us is that it requires an end to add on to. So if we were to draw this, if we had this DNA sequence here and we had another DNA sequence here that was just a little bit shorter, and these would be connected together, if this end was available, DNA polymerase could see that and it could actually add on to it and start making that end longer. But if there wasn't an end there, then it wouldn't be able to just start making new DNA. So it does have to have an end, and even more specifically, it requires a 3' end. So recall that the 3' end has a hydroxyl group on it. We had two unique ends to our DNA molecule, and so if I just draw it again down here, we have a 5' end and we have a 3' end, and DNA polymerase needs to be able to see a 3' end that it can add onto. We are going to have anti-parallel synthesis, so we do want to make our new DNA in the opposite orientation of the old DNA strand that we have there, and it is going to always be DNA polymerase adding on to the 3' end. So we will need to have another molecule to get us started. So if we had this kind of starting point that had already been added on here, and if it was anti-parallel to the template as we see it here, then DNA polymerase will be able to recognize this 3' end and it will extend that. So in that case, this would become my new 3' DNA end. So if we talk about that in another way, or we just mention it in another way, DNA polymerase is going to add to a 3' end. Or in other words, it is growing, extending the 3' end. What it cannot do is it cannot add on to a 5' end. It absolutely cannot do that. So we will always have to have a 3' end available, and this is going to affect the overall direction of the DNA synthesis. So if we go back to what we had before and we start talking about how DNA polymerase is going to be involved in this, the first thing that we're going to have to pay attention to is the fact that we have to have this available 3' end. Okay, so here we are back at our picture, and we need to have, if you look at this DNA sequence that we have, the old DNA, it's DNA there, but there's no 3' end to the molecule that we can actually add on to. So that is the first thing that we need. So this available end is going to be what we call a primer. Okay, so a primer is going to be a short piece of RNA that is really stuck to the DNA template. And so we'll see that actually connected on here. And typically this happens to be about 8 to 10 nucleotides long. I'm not going to draw it that long just because of the size of my picture here, but I'll put a short little piece right here. So I'm drawing it in a different color, and I'm drawing it in purple here, pink here, to represent that this is not DNA that we're looking at. Okay, so this would be my short little RNA primer, and I'm going to have one down here, and I'm going to have another one up here at the top. And when I'm putting this on, I'm making sure that it is following the base pairing rules that we have here, and it's also going to be anti-parallel to the template. So I'm going to put my primes on here, and notice this top strand has a 5' over here, so I have to put the 3' end of my primer there. And same thing on the opposite side. And I can add my primes to this bottom primer also. Now we do have an enzyme that is responsible for actually putting that primer onto the DNA template, so building it directly on top. And that enzyme's name is going to be primase. Okay, so primase, again, ACE indicates that it is an enzyme. Primase makes the primer. Okay, so once this is accomplished, we have exactly what we need here, because when the DNA polymerase comes in and it wants to actually make the new DNA, it's going to look at this and it's going to be able to see that here I have a 3' end and here I have a 3' end. So we've got the ends that we need available to actually start the whole synthesis process.